Students, welcome to the second video module of the chapter molecular basis of inheritance. अगर पिछला वाला वीडियो नहीं देखा है तो जाओ देख के आओ. That was upon the DNA structure and proof that DNA is the genetic material. We are making the chapter as 6 video modules. So this is the second video module, and I request you to see all the videos in order so that you can understand very easily. So let's start. Okay. So today's topic of discussion is replication. So what was replication? Basically replication was the formation of DNA from another DNA. Okay, that is DNA to DNA. Okay, that was the replication. Okay, this scheme suggested that two strands would separate and act as template for the synthesis of the new complementary strands. Okay, so we know as कि DNA होता है एक double stranded molecule. Okay, so the two strands replicate one by one, then it joins to form a new DNA. Okay, the DNA formation is a semi conservative method. Okay, so DNA एक semi conservative method से replicate होता है, and इसका एक experimental proof है which was done by two scientists, that was Matthew Michelson and Franklin Stahl. ये scientist के नाम बहुत ही important है. I will surely recommend you to remember the name of this scientist, that is Matthew Michelson and the Franklin Stahl. Okay, so उन्होंने क्या किया था? So they grew E. coli in a medium containing 15 NH4Cl. So N15 is a heavy isotope of nitrogen but not the radioactive one. Okay, as the only nitrogen source for many generations. As the result was the N15 was incorporated into the newly synthesized DNA. Okay, this heavy DNA molecule could be distinguished from the normal DNA by centrifugation and through the cesium chloride. It was asked many times. Okay, the cesium chloride से हम कर सकते हैं centrifugation. Then they transferred the cells into a medium with normal 14 NH4 cell that took samples at a various definite time intervals. Okay, तो the extracted DNA that remained as double-stranded helices. तो हमको क्या मिला? There is a DNA that was extracted from the culture one generation after the transfer of N15 to N14 medium had a hybrid of the intermediate density. You can see the diagram. Okay, कि there will be one strand that will only contain N14 DNA. There will be strand which they will contain one N14, one N15, again one N14, one N15, then again one N14. So what happens? कि the genetic material or the strand of the DNA, one is from the parental source, another one is a maternal source. Okay, so this is the semi-conservative method of DNA replication. Okay. Now coming to the most important topic, that is how the replication happens. Okay, for this I will take a fresh page and discuss how it is done. Okay, so you know DNA is a double-stranded genetic material, right? So this is a DNA. This is the 3' to 5' end. Okay, and this is the 5' to 3' strand. Okay, so let's discuss one by one. Okay, so basically DNA होता है एक हाइपर कॉइलिंग की तरह रहता है. ओके, तो हमको पहले वो डीएनए को खोलना पड़ता है. ओके, सो वी हैव टू ओपन अप द डीएनए की ताकि रेप्लिकेशन स्टार्ट हो पाए. सो दिस ओपनिंग अप ऑफ डीएनए is done by the enzyme helicase. So this is done by the enzyme helicase that opens up the DNA. Okay, after the opening of the DNA, the strands are held in the same position, right, ताकि वो फिर से coil ना हो जाए. So that is done by enzyme, that is the topoisomerase. Okay, that is topoisomerase. The names of the enzymes are very important in the exams. They give you the question कि the unwinding of DNA is done by helicase, topoisomerase, DNA polymerase. Okay, this kind of questions. Then after the whole thing is done, then we start the replication. Okay, so DNA, always remember कि DNA dependent DNA polymerase that always makes strand to the 5' to 3' direction on the strand complementary to it, that is 3' to 5'. तो यहाँ पे कौन सा तुम्हारा मेन स्ट्रैंड होगा? That is leading strand, that will be this strand. Okay, this one will be leading strand. Okay, so leading strand में क्या होगा? First एक प्राइमर जुड़ेगा. Okay, primer that will be done by RNA primase. Okay, that will be done by RNA primase. Okay, that will be done by RNA primase. Okay, after the primer is joined, the DNA dependent DNA polymerase will start its function. Okay, let's say here is a sequence of DNA. Okay, TGC. So the complementary DNA strand will be produced. Okay, so let's say T, A, C, okay, G, like this, okay. And in the leading strand, the replication will be continuous, okay. The replication will be continuous. But in the lagging strand, the story is a bit different. We make small DNA fragments. Those are called the Okazaki fragments. Those are called the Okazaki fragments, okay. These are the Okazaki fragments. They are small DNA fragments. And with the help of the Okazaki fragments, we do our replication, okay. So on the lagging strand, the replication is discontinuous. That is discontinuous, okay. It is discontinuous, okay. So यहाँ पे क्या होगा कि लेट्स से यहाँ पे जो होगा लाइक टी ए सी जी, तो यहाँ पे उसका कम्प्लीमेंट्री होगा, राइट. यहाँ पे भी उसका कम्प्लीमेंट्री होगा, ओके. सो इट इज डन बाय डीएनए डिपेंडेंट डीएनए पॉलीमरेज, ओके. डीएनए डिपेंडेंट डीएनए पॉलीमरेज, ओके. तो तैयार हो गया स्ट्रैंड. अब क्या करना है? We have to excise the primer and the Okazaki fragments, okay. We have to done the excision, that is proofreading, okay. So proofreading is done by DNA polymerase 2, DNA polymerase 2, okay. And the whole process of replication is done by DNA polymerase 3. DNA polymerase 3 does the whole replication, okay. DNA polymerase 3 does the whole replication and DNA polymerase 2 does the proofreading. So excision is done. Now two different strands are produced, okay. So now they need to be joined, okay. So they are joined with the help of the enzyme ligase, that is DNA ligase, okay. That helps in joining of two different strands, okay. Joining of two different strands, okay. That is done by the DNA ligase enzyme. So this is the basic machinery of the DNA replication. Let's say what they have told. The main enzyme is DNA dependent DNA polymerase, okay. Since it uses DNA as template, so these enzymes are highly efficient enzymes as they have to catalyze polymerization of a number of nucleotides in a very short time, okay. Oh, acha. This Y is known as replication fork, okay. This is known as replication fork, okay. This is done during the DNA replication, okay. The DNA dependent DNA polymerase catalyze the polymerization only in one direction, that is 5' to 3'. What I said, it is only unidirectional, that is 5' to 3', okay. On the leading strand, the replication is continuous, and on the lagging strand, the replication is discontinuous, okay. And the joining of the fragments are done by the enzyme DNA ligase, okay. So basically I have made you understand the topic of DNA replication in under just 10 minutes, okay. And you can almost solve every question of DNA replication. So let's see, you can solve or not. In a DNA molecule, the number of hydrogen bonds between adenine and thymine, it is very easy. Adenine and thymine, they have said the answer will be 2, okay. This was the question from the previous video module. Sorry, it has been misplaced here, okay. DNA replication is said to be semi-conservative because each new DNA molecule contains 2 new strands. Each DNA molecule contains 1 parental strand, 1 new strand. The process occurs only in one direction. It requires RNA primers. So, मैंने क्या बोला था? One from the paternal side, another one from the maternal side. Okay. See, the enzyme involved in the unwinding of DNA strands during replication is. So what I said, unwinding किससे होता है? Helicase से. Unwinding is done by helicase. Okay. Which of the following statements is correct regarding the process of replication in E. coli? The DNA dependent DNA polymerase catalyzes the polymerization in one direction, that is 3' to 5'. This is wrong. This is 5' to 3'. The DNA dependent RNA polymerase catalyzes the in one direction, that is 5' to 3'. This is correct, but they have given the wrong thing. They have taken the RNA polymerase, so this will also not be the answer. Okay. So they have said कि DNA dependent DNA polymerase catalyzes the polymerization in the 5' to 3' as well as the 3' to 5' direction. This is also wrong. This is also wrong, right? So what will you answer? The DNA dependent DNA polymerase catalyzes the polymerization in 5' to 3' direction. Okay. So this is the diagram of a replication. Okay, you need to just remember for the diagram based questions. Okay, this is the template DNA, that is parental strands, and this is the new strands. Okay, red ones are new strands. This is continuous synthesis, this is discontinuous synthesis. You can see very easily. Okay, So yeah, this is all about replication and its MCQs. Hope you can do all the MCQs in the NEET exams from the replication topic they have asked. Okay, and for the DPPs, kindly follow our YouTube channel and please visit our website where you can find more interacting DPPs and the test series. Okay, thank you.